Hi, this is Miss Litton, and this is still period 3, APBio. So now we first laid a firm foundation, right, about evidence that we have a universal common ancestor. What were the 3 lines of evidence that we used? DNA, RNA, and ribosomes, universalness of them. Number 2, shared genetic code, to code for those amino acids. And what's number 3? Metabolic pathways, signal transduction. All right. So now let's talk about how that gets passed on. In order to do that, you always got to start with Mendel. Now let me tell you a little something about Mendel. He did not know anything about chromosomes. He didn't know anything about mitosis. He didn't know anything about meiosis. He didn't know anything about cell structure in any way. He did not know about genes, and he did not know about alleles. Why? Hadn't been discovered yet. There was no microscope for him to look through. Yet he is known as the father of genetics because of his amazing scientific method. How he was able to do his research, how he was able to study and write his hypotheses, how he was able to collect his data and use math to statistically analyze that data. That's why he is known as the father of genetics. If only Darwin could have known about Mendel. They were contemporaries. Mendel read about Darwin, but Darwin didn't read about Mendel. His work, Mendel's work, was not published until after he died, and its significance to the scientific community was not understood for about 50 years after he passed away. This would have been huge had Darwin had access to these materials, because what wasn't understood is before Mendel, traits were thought of as a blending, an amalgamation of your parents. And so that would be like white paint and black paint mixed together forming what? Gray paint. They didn't understand the particulate matter of inheritance, that your mom might give a white allele and your father could give a black allele, that they were independent of each other. They saw it as just an amalgamation, blending. And what that couldn't help support is Darwin's theory of natural selection in the source of his variation. You have to have variation for natural selection to work, right? Because nature is selecting which of the variations is best adapted. Those are the ones that are surviving and reproducing and making more offspring. And giraffe necks are getting longer and longer and longer because having, bless you, a longer neck made it so you could eat the leaves at the top of the tree, not just at the bottom of the tree. But if you're an amalgamation, then you would just get to be the average of everything, yes? So Darwin couldn't explain that because there was no idea about the particulate matter of genes. Now let me tell you a little bit about him. He studied at the University of Vienna, and I just thought you'd like to see their botanical gardens because I think they're really pretty. He was a substitute teacher at one point, and he became a monk. His parents were farmers. This is probably why he started studying peas, as he was already familiar with gardening. And thank goodness he picked real concrete, obvious traits. Like peas are either tall or they are short, right? Is that the way it is for us? No, you're not like this. There's not like 2 heights out there. But for peas, there were. So there are very discrete traits that you could quickly crossbreed and look at the offspring and count. And they wouldn't have like 2 offspring or 1 offspring. They would have 1,000 offspring, and he was able to record and look to see how many are tall, how many are short. We can't do that with humans. I can't go, well, he has dark hair. I wonder if he is a carrier for a gene with light hair. Well, I need to cross him with somebody who has light hair. I see somebody who has light hair. So these 2, go ahead, step into my office, have some sex, and then we would be able to take our data next year when you're in chemistry, right? Do you see what I'm saying? He picked peas, easy to cross-fertilize. I'm going to come back to that, so put a pin in it. He would be able to... We're very prolific in this class. He would be able to study traits quickly, and that's exactly what he did. So prior to Mendel, it was all about blending. This was no help to Darwin, didn't account for variations. Now, due to Mendel, there was the particulate theory of inheritance, accounts for differences and variations. Again, he called what we call alleles now, variations on a gene, he called factors. And so you're going to see me use that word every once in a while. If only Darwin could have known. Now, the peas you eat, if you had peas, maybe you had green beans at Thanksgiving, I don't know. But if you had peas, what color are peas normally? Green. However, yellow peas are dominant over green peas. So, what? But since forever. So now, listen to me for a minute. Just because a trait is the most prolific does not mean it's the dominant trait. For instance, how many fingers do you have on your hand? 5. What is the name of your band teacher? Mr. Borges. He has 6 fingers. He was born with 6 fingers. He's had it removed. It's not like parents go, oh, yay, let's keep that. That's unique. Okay? 6 fingers.
He can play 4 trumpets.
Wait, he can do more instruments.
Yeah, it's not usually as functional.
It's
just
like a skin tag kind of type, or
but larger.
A bigger one, yeah.
But larger. So, come back to me. If you get the gene for 6 fingers, that is dominant over having 5 fingers. What does that tell you about when Mr. Borges, if he decides to have kids?
6 fingers.
Is it for sure? No. If he chooses to reproduce with a 5-fingered woman, then she will only have the gene for 5 fingers. That is a recessive trait. Now, both of his parents were not 6-fingered individuals, only one. So that means he is a hybrid for his 6-fingeredness. So he has a 50-50 shot of having a 6-fingered child or having a 5-fingered child. Now, if he has a 5-fingered child, let's say his first child, Joe, okay?
Joe Borges.
Joe Borges. Let's say Joe has 5 fingers. Does that mean Jeb, who came second, is going to have 6 fingers? No, because Chance has no memory. Every time he has an offspring, it's always going to be a 50-50 chance. Okay, do you see all that? Okay, thank you, Mr. Borges. Now, Mendel, okay, here you can see when he was born, when he died. Mendel used peas. Those were his favorite, really easy to study, real discrete traits that he studied. But here's his little experimental procedure. He used a lot of statistics, used a lot of math, prepared his experiments carefully, and conducted a lot of preliminary studies. This is why he worked with pea plants. Youngest bio buddy, tell your oldest bio buddy why he worked with the garden pea. Okay, so we need to understand what does it mean to cross-pollinate or reciprocal crosses. So youngest bio buddy, you're talking here, but oldest bio buddy, tell them what it means that you can cross-pollinate or reciprocal cross. Go ahead.
So pollinate each other. So there's different. There's a male part and a female part of a flower. You know that, right? Yeah. So that they pollinate each other, which means that.
So not a single sex. Each plant had male and female parts. So the male part of one could pollinate the female part of the other, and vice versa. Okay, cross-pollinate. It doesn't work like that with us, right? So this is easier to get data on because you're not bringing in the male-female part of it. Okay, back to here. He chose 22 true breeding varieties that were very simple, and he kept really careful records, really good data. In fact, some people thought his data was so good that maybe he falsified it, okay, because it was so close, so accurate to what we know today. So he looked at things like tall versus short, and tall was dominant, round peas versus wrinkled peas. Okay, there we go. round peas versus wrinkled peas, and round was dominant, and yellow peas versus green peas, and yellow were dominant. Now, when you cross your parental plant, if you have a true breeding tall plant, that means generation after generation of tallness. And you cross that with the true breeding short plant, generation of short. When he crossed a tall with a short, what was dominant? Tall. All the offspring were tall. Those offspring are called F1s. When you cross F1s with each other, like siblings, but it's okay because they're plants, okay? When you cross those F1s with each other, their offspring are F2s. Now, I want you to look to see what kind of patterns do you see in the F2s. And I'll put this up here. Okay, talk about it with your bio buddy. Go ahead.
Yeah. It's probably
Big numbers, right? But what pattern do you see between dominant and recessive in the F2s? Yeah, 3 to 1. Is it exactly 3 to 1? No, but it is close. Now I'm just curious. If this were 3 to 1, then this would be 224. This would have to be 3 times that, right? So that'd be what? 2, 4, 6. What would it be?
670.
Is it? No. I wonder, is there a test we can apply to see if it's off in a significant amount?
Punnett Square.
Okay. So now what you can do is you can look at your data and analyze. I would predict it to be 3 to 1, but it didn't turn out 3 to 1. Am I off enough where I need to look that something else is influencing the heredity, right? Or is this within an amount that's acceptable? Okay, so we will be doing that. Now, let's write a little bit of notes on Mendel. Okay, background: Gregor Mendel, Austrian monk, studied math and science at the University of Vienna, was a substitute teacher and had farmers for parents. His work went unrecognized during his and Darwin's lifetime. Before Mendel, it was blending. Offspring would have traits in the intermediate between those of the parents. That's what everybody thought. It did not help Darwin because it did not account for variations. Mendel has what's called the particulate theory of inheritance. Traits are inherited as particles. Offspring receive a particle from each parent. Now we would call those particles genes. And we would call variations on the genes, what would we call the variation of a gene? Alleles. Alleles, good. His experiment used statistics, careful and deliberate following of the scientific method, kept detailed, accurate records, conducted preliminary studies. He used garden peas. Why did he use garden peas? This is what you need to put right here. Easy to cultivate, short generation. And then beta note taker, you can pick up B. He chose how many true breeding varieties to look at? 22 true breeding varieties. Okay, everybody good? Does everybody have it? Okay, so I showed you some data here. Here is some more data. Okay, can you see the pattern here? Following pretty close to what? 3 to 1. And here you can see some of those traits. Purple is dominant over white. Now keep in mind, when you crossed a true breeding purple with a true breeding white, all of their offspring were what? Purple. So you would think that the white trait went away. But yet when they crossed it and looked at the quote-unquote grandchildren, all of a sudden the white trait started popping up. And that's what he was looking at to try to understand. That's where he came up with his particulate theory of inheritance. All right. Now, let's try, let's see. Yes. Oh, here, I have a joke for you. Okay, good. Oldest bio buddy, listen as your youngest bio buddy explains this. They will have no trouble explaining it to you. Go ahead.
It's like a 1 out of 4 chance.
All right, now switch. Go ahead.
In a perfect here's a T, short and tall. You get tall that first generation.
Okay, now we need to talk about this for a little bit because look at that tall plant. What do they have up there? They only have one letter for the tall plant. Do you see that? What does it say?
Capital T.
But for the offspring, you see how they have 2 letters? Okay, so they're kind of misrepresenting in this particular model. I want to draw your attention that really you would have a homologous pair of chromosomes, yes? And this one says big T and this one says big T. So that's why they just wrote T, because the only thing this parent had to give was a big T. You all right with that? Okay, now what happens during the S stage of interphase?
DNA replication.
Right? What stage am I drawing right now? What stage? Prophase, and if they come together, it would be synapsis, and then what stage would follow prophase 1? Metaphase 1, right? Then what? Anaphase 1. It doesn't matter. Every single time you're going to get a big T, right? You okay so far? Same thing over here. Now, this is not a monohybrid cross. Crossing this plant with this plant would be a monohybrid cross because each of these are hybrids. This is referred to as a heterozygote. Hetero means different. This one is just, what's his genotype? Little t, little t. That is his genotype. Being short is his phenotype. Okay? So if you cross a true breeding tall with a true breeding short, they're all going to be heterozygotes, big T, little t. This is where we use Punnett Squares. This is where you're going to need some sort of piece of paper or digitally do a little note right now and draw yourself a Punnett Square. Start off by drawing a Punnett Square. Remember, you have a stylus. Now, if it identifies in your genetic cross as male versus female, as you're crossing a male with a female, can you look up here just for a minute? Okay, I don't want to hear about it, but the female always goes on top. So if the female is, I don't want to hear about it. If the female is big T, little t, one of her gametes would be big T, one of her gametes in her eggs would be little t. Are you all right with that? And if the male, big T, little t, then this would be sperm, let's say, or pollen. Now stop and look at me for a minute so I can go over this with you, okay? It is not saying that every coupling is going to result in 4 children. It could result in 1 child. It could result in thousands, right? Okay? This is telling you the statistics. Look at me. Each individual has a homologous pair. Eukaryotes were diploid, right? So I have 2 copies. So 1 copy, 1 copy. And then whoever you're reproducing with, 1, 1. You follow? Because of the law of segregation, you will separate out the alleles. Now, if this sperm right here, all the sperm are here. If this sperm right here fertilizes this egg right here, then you will have the genotype big T, yes? Okay, I'm going to put this back. Go back, sperm. Can't do that in real life. Okay, so you will get the genotype big T, hashtag life lessons. Okay, Karen. All right, you will get the genotype big T. But what will the phenotype be for that individual? Tall. Okay? Fill in your boxes for me. Now, try and write out your genotype and phenotype ratios. Let's see how you do. Don't leave any numbers naked and always round down. I hope you're trying this at home too. Okay, you ready for me? If you're done, poke your bio buddy with your stylus and then compare your answers when you're both done. You didn't do what I asked you to do. Why are you I'm saying words to you. This is where you respond. I'm sorry, I should have done it the way you asked. Okay, come back to me. You ready? What did you put right here? Big T, little t, and then what? Good. What'd you put right here? Now, you could write little t, big T if you're keeping it in order, but it doesn't make any difference. It still tastes the same. Big T, big T. Phenotype is what? And what's your genotype here? Okay, we good? How many different genotypes are there? Show me with your fingers. Come in. Saying 3? Some of you were afraid. Yes, there are 3. What are the 3 genotypes? Yes, 1, big T. You always start with the most dominant. Then what? How many? 2. Then what? Those are the genotypes. Those are the actual alleles on the homologous pair. Genotypes, think genes. Okay, think genes. Phenotype, think proteins. How does this code out in a protein? How many different phenotypes are there? Show me with your fingers. How many different phenotypes? Some of you are still not committing. It's 2. What are they? What are they? How many tall? To what? 1. Discuss with BioBuddy. Why don't those match? You have 2 copies of each allele for each trait, yes? However, one allele could be dominant over another allele. So there are 2 ways to be tall. Big T, and big T, little t, you're always still going to be tall either way. Okay? Now, this showing that out would be something like this. You know, it's the same idea. They're showing you tall and short. I want to remind you here relate this right here to inheritance. If this were an individual, talk to me about their offspring. Not it. Go. Now what? Now, wherever the tall allele goes, so too is the white allele. And wherever the short allele goes, so too is the pink allele. This looks pink to me, but yes, I agree. Unless what happens? Crossing over. Then you could get some more variety. Because you could get a tall plant with the pink, and you could get a short plant with the white. Do you remember crossing over? Okay, when does that occur? Synapsis, right? At synapsis during what stage? Prophase 1, crossing over. And that could mix it up. These are called linked genes. Let me give you an example of linked genes. Hair color and eye color. If you have dark hair, you tend to have dark eyes. If you have white hair, blonde or red, you tend to have light eyes. They travel on the same chromosome. Raise your hand if you have dark hair and dark eyes. Say a P. Okay. Now, raise your hands if you have light hair and light eyes. Okay. Now, raise your hand if you are evidence of crossing over. Now, this hair is blonde, but it's supposed to be brown. Remember, summer, it's the hairdresser, not the season, right? I'm supposed to have dark hair and blue eyes. Raise your hand. Who else? Dark hair. Now, who? Yes. Yours is, because you have red in it, right? Yeah, hazel, but your hair is brown. Okay, then raise your hand. Raise your hand. Okay, now, who has light hair, blonde hair, and dark eyes? Yeah, you have blonde hair. And what color are your eyes?
Brown.
Brown. Okay, so raise your hand. So there's potentially 4 of us in this room who are a result of what? Crossing over. 4 of us in the room that are a result of crossing over. Now, it didn't happen when I was made because my mother had dark hair and blue eyes. And it happened before that because my grandmother had dark hair and blue eyes. So it happened somewhere in my ancestral past. Does that make sense? But linked genes, what if there was a different gene right here? Oh, it doesn't matter. What do you think it's more likely to have crossing over between being round and wrinkled and white or pink, or round and wrinkled and tall? Because there's more what? Space for crossing over to occur. When genes are linked very tightly on a chromosome, there's less space for crossing over. The farther they are away, the more likely you can have mixtures due to crossing over. Does that make sense to you logically? This is how they sequence genes, is by the rate of crossing over to know how far apart they were on the chromosome, is how much they crossed over. All right, so on your notes, take a look at your notes. You have monohybrid cross, and I have that all filled in, yes? Okay, Mendel's law of dominance. Mendel's law of dominance. It says that one trait or factor can be dominant over another. For example, tall plants are dominant over short plants. Okay, and I would like, please, youngest bio buddy, can you explain this diagram right here? And then you're going to switch. Somebody else is going to explain the next one. I need like 3 volunteers to come wash their hands. Just come on up. I don't care. First ones, whoever, because you're going to distribute something. So wash and dry your hands.
The albino.
I said use soap. Okay. Switch. Explain this. Why is this called the law of segregation? What is getting segregated?
Well, if they separate from each other, so it's sister chromatids along with pairs, tetrads together. They make centromeres, right? Because they meet at the mean. And then they separate, right? And so, yeah, so segregation is like a separation.
Guys, look at me for a minute. This is a piece of paper that you're going to receive. It's a long strip, okay? Put it in your hand or on something clean, not on your desk because your desk is dirty. So they just wash their hands, okay? So put it on something clean like your hand or a clean piece of paper. Don't put it on your disgusting Chromebook or desktop. Those are just disgusting. Okay? And I'm going to keep talking to you while they're handing those out. Just keep it, set it aside, don't do anything with it, just hang on to it. Okay, look at me for a minute because I know you can do two things at once. All right. So the first law that Mendel came up with was the law of dominance. There are exceptions to that rule. For instance, you can cross a red flower with a white flower and get pink. You with me on that? That's something called incomplete dominance. Or you could cross a red flower with a white flower and get some red petals and some white petals. That would be codominance. I'm going to talk to you more about that. You could cross a black bunny with a white bunny and the bunny could be what? Gray. So it could be truly every hair is gray. I'm not saying it works like that with bunnies. I'm just giving you an example. Or you could take, and just put them right back in my hands when you're done. You could take
Should we draw one for ourselves?
Yes, you need one for yourself. Josh will get the one he dropped on the floor. I'm just kidding. It's okay.
I'm sorry.
That's all right. I don't want that one back. Trying to teach. Okay. All right. You could cross a black bunny with a white bunny and get a gray bunny, okay? Or you could cross a black bunny with a white bunny and he could have some black hairs and some white hairs, but from a distance it looks like gray. Do you see the difference? Okay? So there are going to be exceptions to the law of dominance. This next law, law of segregation, if you get an exception to this, it will always be a problem. Do you remember non-disjunction? Remember trisomy 21? Yeah. So they will segregate. If they don't segregate, there's an issue. But basically what that means is if you're a hybrid and you have big T, can I get eyes up here, please? If you have big T and you have little t, right? Are you with me? I'm a hybrid. What's going to happen during the S stage? Denarrative. Okay. Law of segregation says in anaphase 1, big T will be separated from little t. So a gamete is not going to get a big T and a little t. It's either getting a big T or a little t. Law of segregation. You know that already because you did a Punnett Square and you demonstrated the law of segregation. When they were big T, little t, you separated out those two alleles. That is the law of segregation. That's what set up that Punnett Square. Okay? Now, we looked at these. These are genes that are on the same chromosome. T and S are linked genes. Do you agree with that? But you notice that you have the same sequence because that's a homologous pair of chromosomes, the same size, the same shape, and they code for the same characteristic. The red one could be from mom, the blue one could be from dad. You okay so far? All right. Now, when somebody has genotype versus phenotype, eye color is not a discrete thing like how pea plants are tall or short. Eye color is not like that. We oversimplify when we say dark eyes or light eyes, and dark eyes are brown or black, and light eyes are blue or green, right? That's an oversimplification. There's like 14 different genes that code for eye color, just like intelligence. It's not like you're smart or stupid or semi-smart, right? There's a lot of different genes. That's a polygenic trait. Multiple genes code for that trait. But brown eyes are dominant over blue eyes. Okay? So you can have brown eyes, you can be big B. Big B, that would be called homozygous, or you could be big B, little b, and that would be what? Heterozygous. This would be homozygous recessive. This is homozygous dominant. Yeah. Now, I'm going to tell you, I used to do a lab in honors bio where you took all your traits and then you went and looked at your parents and then you saw and predicted whether they were dominant or recessive. Yeah, I stopped doing that lab. It's multiple years ago. He sat right there in that chair on the lab bench. He was an outspoken, well-loved boy, and I bring up a trait that's dominant, and they're writing it down and trying to predict from their parents who they got what from. And he goes, Miss Linton, I have brown eyes, but my parents have blue eyes. And I'm like, oh, well, there's 14 different genes. There's different ways our gene can be masked. We get to the next trait. Oh, Miss Linton, I have my parents have I'm all, well, could be, I kid you not, 4 different times his hand goes up. Pretty soon it just became #awkward because kids are like, every time he raises his hands, they're like, like, what didn't happen? A talk had to happen. A talk had to happen because there was no way that could have been from those parents. Okay? Yeah. Yeah. I didn't ask how the talk went. Now. So I am testing you on one thing, but one thing's not going to prove anything. That's what that paper's about. We'll get to that paper in just a minute. Hang on to that paper. You think, I'm adopted. No, not one paper. Yeah.
So I have green eyes, but like my dad's brown eyes. So does that mean? So does that mean?
Yes, Josh, you're adopted. Yeah, he's a carrier for that recessive gene. Yeah. Okay, now, widow's peak. Okay, widow's peak. Now, for some reason, people don't like to say they have a widow's peak. I don't know why. It is a dominant trait. It doesn't mean it's the most prolific here in this classroom. But who's this? Do you know who that is? It's Lance Armstrong. He has a widow's peak. Do you know who this is?
Ice Cube.
It's not Ice Cube. What? Jamie Foxx. He's a very good piano player. He has a straight hairline. Do you know who this is?
Jamie Foxx has the same chiropractor I do.
Oh, do you know who that is? Vince Vaughn. Yeah, he has a widow's peak as well. Show your hairline to your bio buddy. Do you have a little widow? A little widow or a straight? No. Now that's something you can go home and check. Widow's peak is dominant. Okay? Now, take a look here. Let's look at these. Let's decide what are the possible gametes each of these individuals could make. Okay, so take a look up here. Here is a genotype. How many different gametes could he make? Here's a genotype. What would that be? Yeah, big W. That's the only thing it has to give is big W. Do you agree with that? Okay. How many different gametes could this genotype make? What would they be? Yeah, we don't know which one. Chance has no memory, right? How many different gametes for this one? 2. What are they? How many different gametes for this one? What is it? Okay, now decide on these A through E's. Does that represent a gamete or a genotype that you see right here? A gamete or a genotype? Discuss it first with your bio buddies.
No, like, you can have one X.
Okay. Gamete or genotype? Gamete. There's only one D. The genotype could have been this, or it could have been this. Do you agree? Yes. Okay. Is this a gamete or a genotype? Genotype. How many gametes could it make? 1. What would it be? Yes. Gamete or genotype? Gamete. And if it were a genotype, it could be this, or this, right? Be careful when you use letters like P's and W's because bigs and littles look a lot the same. S's, so put a little mark. Like when I do little W's, sometimes I put a little curly Q on the end of the little W, okay, to help differentiate. How about this, gamete or genotype? Do you agree? It's a gamete. The genotype could have been this. It could have been a lot of things, but that's one of it. It's not. You'll be able to do crosses like that between two of those. No problem. Easy peasy, lemon squeezy. Okay, what about here? Gamete or genotype? What? Genotype. And the gametes could have been what? Big P or little p. Good. All right, time to talk about that PTC paper that you got in front. Not PCP. Okay. What you want to do is you only need like a quarter of it or a third of it. The reason why I say that is so you can take home the remaining bits and divide it and test your parents, your siblings if you want to. So rip off like 25% or a third off the end of it. And then you want to save the rest. Okay, now do you have a third or 25% in your hand, holding it in your hand? The other hand's got the big piece, yeah? Take the little piece. On the count of 3, we're all going to put them in our mouth. I've done this a million times, so I know what the answer is for me. Go ahead, and on the count of 3, we're going to put it in our mouth. That's why we washed our hands. 1.
Wait, what do we do with it or not? Like,
Don't go like this. Don't go. 1, 2, 3. The little piece. You either taste it or you don't taste it. And if you taste it, it's bitter.
I taste it.
Yeah, and you can spit it out if you need to. If you can, some of you where it's like, oh, I just put a piece of paper in my mouth. It's no big deal. But for others of you, yeah, that's what's happening. It's like, it tastes like somebody sprayed perfume in your mouth. Go spit it out. Go spit it out.
on the tip of my tongue. Wait, so what does it mean? It felt like
Okay, come back, come back. All right, so if you have a receptor for PTC paper, that chemical, you will taste it. If you don't have a receptor, you don't taste it, right? Who coded, what are receptors made out of? Proteins. Who coded for that protein? DNA. Now, tasting is dominant over not tasting. So if you taste in this room, what does that mean? It's a dominant trait. What else does that mean about where you came from? One of your parents has to be a taster, right? If you are a non-taster, does that mean your parents have to be a non-taster? They could have been what? But they were what? They were what? Yeah, they were a hybrid, a mix, right? A heterozygote. All right, think about, I want you to log in. Log in as something that tasted good from Thanksgiving break.
Wait, so what if I was just for Thanksgiving break. What did you have? Tears? Sandwich, turkey sandwich.
Nobody tasted Krista Hong. Okay. Okay. Something that tasted good over spring break. I mean, Thanksgiving break. Sorry. Thanksgiving break. Maybe something from your Thanksgiving dinner. I don't know. Okay, I'm going to show you the questions so you can work on them. Here's question 1. Here's question 2. Here is question 3. I'll make that bigger for you. Give me just a second. Okay, so the dad is big T, the mom is little t, the children are big T, little t. Okay, and here's the question. pause it if you need more time. Here's question 4. Here's question 5. Here's question 6. Here is question 7. Here is question 8. Question 9. And question 10. Okay, one member from each team, I want you to capture this as your debrief. So take a picture of that and throw it in your notes. So you can, and you're going to do your debrief in your lab notebook. You're going to call it prep for today's date and practice problems 2. Or you can all capture it, I don't care. That is your, you don't have to do no question due. And you don't have to write the question, just do the work to see that you understand it. And we'll check it next time. Did everybody get it so I can go back to the other? Anybody still need it? Okay. Everybody got your debrief? Okay, I want to give you feedback, so I'm going to stop it and I'm going to let you click through, but we will talk through them again next time. So take a look and see how you did. Struggled a little bit on 3. Struggled on 3 and 8 and 9. Let me just hit 3 real quick. What is the genotype of the children? Heterozygote. Okay, and then 8. Could 2 non-tasting parents have a child that could taste? No. Non-tasting cannot have unless there's a mutation.
Welcome to the first day of Awareness Week. Out on the great lawn, we have some mindfulness.
All right, have a good rest of your day.
In the earth.
Hope that was helpful.